But for now, I would like to give the floor to Yolanda and Philip to talk about the Moonshot Restoring Immune Health.
Thanks, Thales. So I'll be starting first. And next slide. So again, I also use the same analogy that Thales started with when I'm discussing with patients when they have an autoimmune disease. Indeed, the immunity is a bit too strong and leads to the disease itself. And what we are usually trying with treatment is to kind of get it into balance. But I think it's important also to realize we don't really get it into balance. It's still kind of almost flipping all the time. So you see that there's also, for example, when there is an infection or vaccination, people can get a flare of their autoimmune disease. So it's always kind of a balancing act. And if we go to the next slide, then you can see also there are several consequences of this disbalance, you could say. But first of all, the very obvious one, in some patients, we just don't get them at all into balance. So they have just an insufficient treatment response. And obviously for them is specifically of most importance to get new treatments that are better than current ones. But increasingly also appreciated is that in some patients, we just don't know whether there is immunological disease activity happening while we are treating them. And that can also sometimes lead to ongoing, slowly progressive tissue damage that leads to finally to disability. This imbalance, in a way, leads often to chances of flare with the corresponding anxiety in patients, and again to the same problems of increasing tissue damage with every flare that they get. And this is probably also one of the reasons that both patients and doctors often try to play it, let's say, on the safe side. So what you get is that often we are over-treating sometimes even patients, and that leads, well, not infrequently to adverse events that might be even prevented. What it certainly also leads to is to very high healthcare costs. I mean, the cost for treatments in autoimmune diseases has been increasing every year. New, more targeted treatments are coming, some of them hundreds of thousands per year. And especially then, it's even more important to surely know that you're not over-treating, but preferably you would also step up from chronic treatment and go to treatments that can actually lead to cure. So when we go to the next slide, you can, if you look at the current treatment, this is very high over, but what we often, you can say they are either targeted somehow on often downstream effects, and I think the complement inhibitors, for example, is a very good example of that, or they're relatively untargeted, like the corticosteroids or a full B-cell depletion. So you're not actually only targeting the relevant cells, but all different subtypes as well. And I think we should be also honest that most of the treatment we use, again, is just maintaining this balance somehow, but then they're not really inducing long-lasting remission or cure. So this is also the next slide. So the approach that we have in this Moonshot, first of all, we would like to repurpose the existing treatments. We know there are a lot of treatments, and some of them can be also certainly used for other IMiDs. We could do this by trials using the same methodology, or even basket trials. But we could also think about stratification of patients by their immunotype rather than their clinical syndrome. On the other hand, we feel that we have to discover also new targets to allow a complete new generation of treatments, so we can also try and get the cure rather than just keep the chronic inflammation somewhat in balance. So if we go to the next slide, I think this is Yolanda.
Yes, thank you, Philip. Yeah, here we go for the Moonshot Restoring Immune Health. We divided the long-term objective, which is develop drug repurposing and our labeling extension strategies and innovative immune-based intervention across diseases, into 3 parts. So partly we want to establish a disease overarching translational framework in 2030. Then we go to 2035, and then the aim is to develop immune modulatory interventions also across IMiDs. And in 2040, we aim to have an application of immune modulatory intervention across multiple IMiDs. Importantly, during the Moonshot is that we do disease overarching research, and it should be collaborative research strategy. Patients' involvement is of course very, very important to ensure also the societal impact, so that it's in the end also for patients of benefit and that they want to undergo these kind of treatment. And the proactive valorization strategy for economic impact is also part of this Moonshot, as it is also for the other Moonshots. Then on the next slides, we go back to Philip.
Yeah, this is a very interactive presentation, as you can see. So in this slide, just a few examples to start with in terms of repurposing. So rituximab, as you all might know, it has been out there now for 20 years, but only now we are actually using it in some of the neurological IMiDs. Multiple sclerosis, obviously, ocrelizumab has been used for many years very successfully, but now there is a large trial comparing it to rituximab. As well, the obvious advantage that rituximab is like almost 10 to 20 times cheaper. But we are also using it in a rare inflammatory neuropathy now to induce cure directly from the beginning, again, to prevent chronic treatment with mainly intravenous immunoglobulins. And also we are trying to set up the same trial with the same trial design in myositis. Another extremely promising field is the CAR-T, the CD19 CAR-T that you might even have a chance to read it in the newspaper in this weekend. But this is, as you know, there have been a couple of patients treated with CAR-T leading to a full remission. And we would really like to know whether this is something viable and can use in both rheumatological and neurological diseases. We are very happy to have obtained funding for this to hopefully provide this proof of concept that this is something that we can treat and also even cure patients. Next slide. Another thing, of course, is to do, and this is also what Tala kind of described, to step away from the organ-based taxonomy, but also future on the stratification. So even when thinking about future basket trial, you would rather not select patients on their clinical syndrome only, but on the presumed main or dominant pathomechanism that is underlying the disease. So this is also something that we have been doing for years, mainly on the cytometry to recognize the immunotypes that could also help us choose patients for the appropriate trials, because we know that one size will not fit all for all diseases probably. And even if you have the luxury, and again, you might be getting worried by all the neurological examples, but I'm neurologist at the end. But even in MS, where there has been like a revolution in terms of treatments that you can choose from, then you still don't know when you have a patient in front of you which to choose first, because actually we don't have predictive biomarkers to help us with this. And finally, there are also a lot of rare diseases like CIDP, for example, where it's, well, surely impossible to do the big trials that we are kind of used to doing some other diseases like rheumatoid arthritis and MS. I think the next is yours.
My turn, yes. Also want to show you an example of what we did in the previous projects. And this was, here we made use of PLGA nanoparticles. We got advice from the CBG that this kind of therapy can be called immunomodulatory therapy. So, and why do we choose PLGA? That's because PLGA is approved by the FDA, and we can get it, we can apply it directly in vivo in patients. So the quality of the PLGA is sufficient for that. So that's why we choose PLGA. So on the left and on the right, you see 3 boxes. And these are the boxes which we mean, what we mean with activating nanoparticles, immunomodulatory nanoparticles, and tolerizing nanoparticles. This project was originally started as a cancer vaccine. So then we used the activating nanoparticles, in which we brought an antigen and a stimulatory adjuvant into a particle and vaccinated today 9 patients. And we wanted to get a readout of course, antigen-specific T cells recognizing, so cytotoxic T cells recognizing the cancer type. We also developed in one of the previous projects immunomodulatory nanoparticles. For this, we used, for example, PGE2 receptor antagonists, so to decline the working of inhibit PGE2 from executing its function. PGE2 is often very high in cancer patients, but also in patients with suffering from an immune dysfunctioning, so in IMiDs. And that would also be helpful to be able to downregulate this often the very bad action of PGE2. Furthermore, we also developed tolerizing nanoparticles, and for the tolerizing nanoparticles, we used vitamin D3 or the peptides, and then we hope to get, instead of a cytotoxic T cell, what we aimed for by cancer, we hope to get regulatory T cells recognizing very antigen-specific, one of the antigens what is causing the chronic inflammatory diseases. This project on the tolerizing nanoparticle part is still in the in vitro phase, so it's still only in the we are only performing experiments in vitro, so for rheumatoid arthritis and diabetes. And since we know when the tolerizing particles can tolerize actually or induce regulatory T cells, we can exchange the peptides, the antigen, for the disease-specific peptides of that disease. So what we aim for is that the dendritic cells are targeted in vivo by these nanoparticles. This can also be done, for example, by tagging monos or DC nanobodies or antibodies to the nanoparticles to get them more specifically into these dendritic cell subsets. So in the end, we hope that we can stabilize the immune balance. Here depicted in a bit different way, but of course we want from activating to tolerogenic in chronic inflammatory diseases and from tolerogenic to activating in cancer. So if we go to the next slides. And so to Philip.
Yeah, so this is the final example. So this is, I think, a truly an example of a disease overarching research in a way. It was during the COVID time in which we included thousands of patients with dozens of IMiDs to study the effect of immunosuppressive therapy on the vaccine responses against SARS-CoV-2. And maybe you can go further. I think there's a second part and another one. Yeah. So what we did basically, of course, we looked antigen-specific T cells and B cells and looked at the effect of in this case, for example, the anti-TNF therapy that was also inhibiting the B cell differentiation and mainly the formation of active B cells. But one of the nice things that we also discovered, and you could also go to the next slide, was that we could also identify these activated B cells using generic markers that were harboring the antigen-specific B cells, which kind of led also to the hypothesis that we could use these generic markers also to track and find the autoreactive B cells in autoimmune diseases. So we looked also, following this hypothesis, we looked back in a study that we did in one of the PPPs just before ImmuneHealthXL, where we did profound site of analysis in different diseases, and you can go to the next, I think, where we looked at myositis and myasthenia, and we indeed could identify these activated B cells between these two IMiDs when we compared it to the controls. So this is obviously, there was a very small number of patients, and we are validating and reproducing these results, but it does give us kind of the proof of concept that we could maybe focus more targeted on specific B cell subtypes rather than depletion of the whole B cell subset. And that, of course, could be used both for maybe developing monoclonal therapies or even new constructs for CAR T. So I think with these examples, Tale, we can give you the floor back for the last part.
Yeah, thank you very much. So I think we've got a nice overview of projects that have been running in the past and examples for future projects. So I think it's good to highlight sort of what's in it for you. We've got now a funding opportunity. Of course, there's for small, medium enterprises and researchers, there's subsidy available. But money is not the only gain that you get. And I think what is essential that is here that you collaborate with a national network of immunology experts across multiple IMIDs. And you can build upon years of expertise and technologies and infrastructures which have been built up over the years and are available and are accessible in that sense. And for, I think, small companies, it's great to have an opportunity to establish proof of concept across multiple diseases for sort of the products that they are developing or prototypes that they want to test in multiple diseases. So besides the funding opportunity, there's really also the opportunity to collaborate and to really also use the expertise around in this network of collaborating partners, not just academic researchers, but also companies which have been participating in past and previous projects. So what is up next? So for the specific funding opportunity, the PPS program, we are submitting the application this Friday is the deadline. The expected outcome will be in December. And once approved, which of course will happen hopefully, we will open in January a call for invited pre-submissions. So do feel free to interact with us to see if there's opportunities there already. And then the following year, we will open another call open to everybody. In addition, within ImmuneHealthXL, we are continuously scoping for new funding opportunities, and you can think of, for instance, the NWO consortia, but also European projects within Horizon Europe or the IHI initiative. But also do think of bilateral collaborations or multi-party public-private collaborations where you see an opportunity. Do reach out and we can sort of connect you to the people who are the experts within the field. If required, we might be able to support any applications or facilitate in the networking and ensure that you and your collaborators have the highest chance of succeeding in a funding opportunity.